For decades, the search to understand Alzheimer's disease operated as an archipelago. The research was characterized by isolated communities, making profound discoveries that rarely connected to a broader map. This timeline begins in 1985. That year, scientists demonstrated that Alzheimer's involves catastrophic cell loss in the locus coeruleus, a brainstem nucleus governing arousal and memory. Concurrently, a separate group of researchers established that microglia act as active immune mediators within the central nervous system. A third discipline documented perineuronal nets, matrix structures that lock mature synapses in place and restrict plasticity. Each observation was structurally correct, yet it took until 2025, 40 years later, for these three biological components to be integrated into a single clinical framework, the collapse trilogy thesis. Why did a comprehensive model of Alzheimer's require 4 decades to assemble when the constituent parts were already sitting in the published literature? The gap is not explained by a lack of sequencing data, computing power, or clinical technology during that period. The delay was a structural epistemic failure. Strict disciplinary silos, reinforced by rigid funding structures, paralyzed neurodegeneration research and prevented the synthesis required for a cure. In 1992, the amyloid-cascade hypothesis proposed that amyloid beta deposition was the single proximate cause of Alzheimer's disease. Offering a solitary causal mechanism, it rapidly became an inescapable citation gravity well. This hypothesis captured institutional funding. The National Institutes of Health structured requests for applications around it, establishing amyloid as the obligatory target. This chart tracks NIH obligations to Alzheimer's projects from fiscal year 2000 to 2020. The concentration is absolute. Projects targeting amyloid and tau absorbed 67% of all available funding, with the amyloid bar crossing $10 billion. By contrast, upstream mechanisms like locus coeruleus degeneration received fractions of a percent. Amyloid dominated the budget for a specific, pragmatic reason. It forms a physical, end-stage lesion that a pathologist can easily see. This created a pathology trap. The field heavily biased its resources toward physical structures that scientists could verify by looking through a standard glass lens. This institutional preference hardened into a self-reinforcing equilibrium. Specialized grant requests and restricted peer review calls reliably rejected alternative mechanistic theories. When Phase 3 clinical trials targeting amyloid, such as bapineuzumab, solanezumab, and crenezumab, failed to produce clinical benefit and were halted for futility, the consensus held. Even after a final wave of BACE inhibitor failures actually worsened cognition, the field chose to refine the exact epitopes and timing of the amyloid hypothesis rather than abandon the premise. $10 billion of concentrated funding did not map biological reality, it mapped what was easiest to measure. Researchers operating outside this consensus did not form a united opposition. Instead, the alternative research landscape was a fractured archipelago of isolated subdisciplines. To quantify this isolation, we can look at a bibliometric overlap analysis of 135 independent submissions to the Oskar Fischer Prize, a program that solicited non-consensus Alzheimer's hypotheses. This heat map displays the phase overlap matrix for that entrant corpus. Researchers within their own domains showed high internal cohesion, represented by the bright diagonal line.
But the off-diagonal cells exposed the disconnect. Cross-pollination hovered between barely 9 and 21%. These different disciplines published in completely specialized journals. They attended entirely separate conferences, ensuring they rarely crossed paths. Distinct training pathways and vocabularies meant these scientists almost never sat on each other's grant review panels. The biological information required to reverse the disease existed in the literature. The institutional channels required to assemble that information did not. The 2022 Oskar Fischer Prize acted as a natural experiment, deliberately structured to bypass traditional NIH review committees. The prize solicited mechanistic models from the global community without demanding prior commitment to the amyloid framework. This generated a massive dataset, 151 independent investigators, working globally without coordination, submitted their models. When the top submissions were audited, a clear pattern emerged. 25 of the 30 graded entrants independently converged on the exact same downstream mechanism. That central nexus was VATPase disassembly, a catastrophic failure of lysosomal acidification. Crucially, these researchers arrived at this exact failure point via completely different biological starting lines, ranging from bioenergetic deficits to lipid toxicity. Others traced the pathology down to this exact same failure point through diverse viral, environmental, immune, and distinct genetic triggers. These scientists did not consult one another. They did not share a common vocabulary, and cross-comparisons show they frequently held contradictory positions on auxiliary issues. False convergence in science happens through coordinated citation cascades. Uncoordinated convergence on a single downstream target only occurs when the underlying biology forces it. The 40-year delay in assembling this mechanistic model represents a massive, quantifiable debt in human health. This debt is paid by the millions of patients currently in hospital wards, relying on a research apparatus that effectively stalled out for decades. If these siloed scientific communities had been placed into sustained, structured dialogue by 1995, the tripartite thesis could have been fully assembled by 2005. This data table models the human consequence of that delay. Looking at the central scenario, the lack of integration translates to an estimated 24.4 million cumulative person-years of avoidable cognitive decline. Preventing this from happening in other fields requires structural reforms that are specific, defined, and completely within institutional reach. First, organizations must mandate that at least 1/3 of grant review panels be drawn from outside a submission-specific citation network, ensuring objective evaluation rather than paradigm enforcement. Second, funding agencies must issue mechanism-agnostic request for applications. These should reward clinical endpoints, like slowing cognitive decline, rather than demanding adherence to a specific protein model. Third, we must build open, machine-readable synthesis platforms. The burden of cross-disciplinary integration must rest on the institutions funding the science, not on the heroism of individual investigators. Disciplinary silos are a predictable design flaw of modern scientific funding, but as the Oskar Fischer Prize demonstrated, they are a flaw we now know exactly how to fix.